All right, I'm recording. Okay, we left off here, looking at the idea of direct counts versus viable counts. So you've done viable a couple times in lab with the idea of the spread plate technique, where you took soil samples, serially diluted it, and then spread it out onto agar plates. And so that's certainly still a good method, because you get to see what's in your sample, and if you want to work with them, obviously you need to grow them up. But it always underestimates what you get through other methods, especially if you do flow cytometry. You get way better population estimates. So the viable count is still publishable data. It's just you have to recognize it's only a slice of the pie in terms of who's there. So when you're looking at who's there, there are fluorescent tags that you can use. You can tag genes, for example, and there's different ways to do it. There's the so-called LAC-Z gene. You'll see that a lot in the published literature, where they say they use the LAC-Z gene. And what happens is that codes for this enzyme, beta-galactosidase, and then you add in a chemical to the microbe's environment called XGAL. So I would do these in Petri plate experiments. And if the bacteria has the gene tagged into it, you can see this blue pigment come up right around the colony. And so it's a very easy visual. A lot of microbiologists, that's how they tend to look for things. They'll use visual assessments of what's happening. And so they usually use some kind of chemical that can change color, and then it's very easy to pick up. Bioluminescence is another way to tag things using the so-called Lux genes. So those are just the genes that allow for bioluminescence, and you can insert them into your microbes and look for microbes. So for example, if I think I came up with this wonderful biocontrol agent and I want to do a field study, and then I can either put the LAC-Z gene in or the Lux genes, release it into the environment, see if it really can form biocontrol, and then take samples periodically and ask the question, is this biocontrol agent even surviving when I put it out in the soil, let alone can it reduce disease levels? And so I would go periodically and take samples, and then in the lab look for either the blue pigment with XGAL or bioluminescence, and that would tell me, yep, my microbe is still in the soil, I can detect it. And so it's a nice, simple, relatively simple way. And then probably the most recent thing is the green fluorescent protein that is being used from jellyfish that you can insert into microbes. Microbes are very relaxed about accepting genes from very diverse other organisms. And so it makes it quite nice to be able to track microbes using just a visual tool. So it makes sense to do some kind of tagging like this when, like I said, if you are asking, I want to put this microbe into the environment and see what happens, and track it over time and see if it can persist in the environment or not. And it's a way to assess that. It's a nice strategy. So under visually, some things that you might see with the green fluorescent protein being shown here, it's very bright under the microscope. And so here you have some plant roots that are showing the nitrogen fixing bacteria in both cases. Here you actually can see a beautiful infection thread that we talk about in plant phys of the nitrogen fixing bacteria getting access into the root hair and making their way to the bulk of the root so that they can set up camp and hang out and fix nitrogen for the plant. Okay, so really nice techniques. And surprisingly, the green fluorescent protein works really well, even though it was isolated from jellyfish. It's been very, very successful in the world of microbiology for tagging microbes. One of the big questions you might want to ask are my samples, who's living, who's dead? And then you can use specific stains to detect the process of respiration. And so there are certain stains you can add in that will pick up on that. Acridin orange, and then the INT stains are used for that. Again, I'm not really asking you to memorize what stains do what, more that you just understand there are stains available that can detect respiration. Here's another one, which is, in my opinion, rather difficult, but it's a little older method. You can incubate your sample with nalidixic acid, and they gave it the term the direct viability count because you do it under the microscope. You don't try to grow things up on a Petri plate. And so you take your sample and you bathe it in this nalidixic acid, and then you let it sit for a little while, and then you look at that under the microscope. And really what you're doing is the nalidixic acid will promote the cells to go into cell division, but it arrests it. It doesn't allow it to go to completion, so it kind of kicks off the process of cell division. And with microbes, when they are starting this process of, when they make the decision, so to speak, okay, I'm going to go through cell division, one of the things they have to do is physically enlarge their cell. And so the cells will get bigger or elongate, and that's what you're trying to pick out with the microscope. You're looking at bigger cells, and that's a tip-off that they are alive and they're starting to go into cell reproduction. It's tricky. You really have to know your microbe, what it looks like normally, and then be able to pick out that it's getting bigger. That can be a little tricky. And then if you have microbes that are really slow growing, this nalidixic acid treatment, if you don't let it go long enough, you'll miss a lot of the slower microbes that kind of just take their time. So this method is kind of fraught with some risks. And like I said, it's an older method, but it is something that can be done. That would not be my first choice, but the epifluorescence is something that is much more standard these days. It's a little bit, it's quite a bit more, it's easier and the results are easier to pick out. Another method that you can do is using autoradiography, which you're using radio-labeled nutrients, which can be dangerous. In fact, our biology department, when I was first hired, I was the radiation safety officer, where I had to go around with Geiger counters and measure for any kind of radiation leaks in any of the labs that were using any kind of radiation for this kind of experiment. And it got to the point where we all recognize there's other tools out there to do experiments that can answer the question in just using safer tools, safer methods. And so we've done pretty much totally away with this idea, but it's still used, and but it is a risk, especially to the scientists, because you're dealing with radiation. So yeah.
On that one, the auto radio.
Yeah.
So how could you tell if something was alive or dead with that? Would the alive ones pick up the free radicals and then you'd be like, okay.
Yeah, because you feed. So what you do is, like when I was doing experiments for my PhD, I would feed radio-labeled phosphorus into the microbe's environment, and they would pick that up and then incorporate it into their DNA or organic molecules. And so then you could use autoradiography to pick them up. You would say, yep, they took it up because they're definitely giving off. I'll show you some pictures of what it would look like. And okay. So it's basically you tag it's usually phosphorus is often what you tag with something that can radiate energy. It's just the energy that it gives off is pretty potent. That's why it's dangerous to us, because it's like getting X-ray exposure. It's kind of like that. And so, like when I would do these experiments, I'd have to wear the full leaded apron, like when you go to the dentist and they, X-ray your teeth. The technician,
they wear that vest.
They wear the vest, and they usually step out of the room before they push the button. That's kind of what I had to do. I had to wear the full, lead vest, and, lead, sleeves, and sit there, and I literally was sweating because you're, essentially getting bombarded with really potent electromagnetic radiation. And so you just hope that the vest is doing its job. It was not a fun experiment to do. And then if you didn't do everything precisely, of course, you had to be very careful with all the chemicals. You had to make sure you didn't spill anything, and they were liquid. So, as your hands are shaking because you're so nervous. I did not, obviously you can tell, I did not like working with it, and I found it very disturbing myself. So I did it once or twice, I should say. And then it's like, okay, I'm done with that. I'm not doing that anymore. Here, looking at acridine orange idea, where the live and dead cells are showing, oh no, this isn't the acridine orange, sorry. This is a different viability stain. But you can clearly, unless you're colorblind, this would be difficult if you're colorblind, but the red, green, you can pick out quite readily under the microscope in this case. And so, the epifluorescence is definitely a lot safer and a lot less expensive as well. Radiated chemicals tagged with radiative molecules are quite expensive. You've probably heard of FISH, which stands for fluorescent in situ hybridization, which basically is just you are creating a probe, a small stretch of DNA as a probe, and then you label it with this fluorescent tag. So if you're asking the question, hey, I wonder if in this sample, do I have any microbes that are antibiotic resistant to whatever antibiotic you're curious about? Then you could look, you'd have to understand what the antibiotic resistance gene is. You'd have to know its sequence, and then you'd make a complementary copy to that and tag it. And so then you could put that into the system and run the experiment, and the probe would then complementarily bind, to the DNA of the organism if you run the procedure the right way. So FISH is looking at, it's a pretty powerful technique because you can really ask a lot of questions using this technique. So it is a favorite tool of a lot of scientists in the microbial ecology realm, but even very applied things like the food industry or looking for certain pathogens in the clinical microbiology world. And so when this technique got whoever invented it, I don't know who, but that was a pretty slick idea. When they came out with this technique, it really allowed for some very nice questions to be asked and answered in a very quick manner. It didn't take that long, and it wasn't super expensive. And it's pretty easy to see the results because of the fluorescence. So you can usually pick out a positive versus a negative test. So let's just look at some pictures here. So on the left, these are the same exact image. It's just the left is without any fluorescent staining under the microscope. What do these cells look like? And on the right, you have the fluorescence. And so, as you've looked at microbes a little bit under the microscope when you did your gram stain. It's really hard to tell them apart because they look so similar. Most of them are rod-shaped or coccus-shaped. It's really hard to tell them apart. But certainly by staining, you can tell there's definitely different organisms here, even though, on the left, the big ones you'd say, oh, all the big ones are probably the same thing. Nope, because when you look over on the right, they stain very differently. And so just going to show how powerful this technique can be with fluorescence. And it really helps the field of microbial ecology because, normally when you look at it under the microscope on the left, it would be very easy to say all the big cells are the same thing. And then on the right, you get the surprise, no, that's not the case. And so these techniques are super powerful for looking at certain questions in microbial ecology. Again, just look at that, all the different colors. And again, under the microscope without the staining, so many of the cells look identical to each other in size and shape. And so again, this is sewage sludge that they were looking at, and they were targeting nitrifying bacteria. And then we have a B is showing a confocal laser scanning micrograph. And so we have a confocal microscope down on the first floor in our building that can do these kinds of experiments. And one of the nice things about the confocal is you can actually, if you do it right, you can keep your cells alive and watch what they're doing over time. And so it's a very powerful microscope technique. A couple of my grad students have used it for some of their questions when they were looking at their microbes. And then another one that's more recent is kind of a tweak to the FISH procedure. They call it CARDFISH. And you don't need to know what a CARD stands for, but if you're interested, it's catalyzed reporter deposition. It's just kind of a tweak. It improves what's the FISH procedure, and it really does allow for some nice questions of gene expression. So I want to know not only who's in that habitat, but I want to know what are they doing. And so gene expression patterns you'd have to look for. So just saying, yes, I see that there's Streptomyces bacteria in this ag field that are doing, I'm hoping they're doing biocontrol by turning on their antibiotics. And so a lot of the Streptomyces I work with, we think that they're using antibiotics to kill off plant pathogens. But just because they're in the environment doesn't mean they're necessarily doing that. And so this CARDFISH technique would be a way to look at gene expression in these microbes. And that's what you want. You want to see that they're actually have those genes turned on and are actually active. So the CARDFISH technique is sensitive to that. And again, it's a great way to ask questions about what is actually happening. What are they doing in the environment, not just whether or not they're present or absent. So this is a CARDFISH. What you would see at the end of this particular experiment, they were just looking for archaea cells. They were just asking the question, how many archaea are present in this sample that they were looking at? So again, very obvious difference between the blue and the green. And so it's a nice visual. It's safe. You're not getting bombarded by radiation. You just have to follow normal safety protocols. Okay. This is kind of a big comparison of several different techniques. And so this I just kind of put in there to get you thinking about what could you do when you go out to a field site as a microbial ecologist, when you start taking samples and come back. You can see the CFU, if they're doing that, that means they're doing a viable plate count, where you end up with colonies on the plate. We call them colony forming units. So this first column is a viable plate count, which is totally fine to do, but you do underestimate. And then you can see the very first column, these numbers are times 10 to the third essentially. But if you look at direct counts, you see your numbers go up compared to the first column. So again, direct counts, you get better representation of the actual population. Because look at that, it's 100-fold difference, 10 to the 5 difference in how many microbes you ended up in these different samples. So again, showing direct count is better in terms of a better way to assess population. And then autoradiography, they use that in order to just say how many are alive and active. So they were using that method to generate that data. And then they could look at their percent of active bacteria versus dormant. And then another easy, fairly easy method is to do some experiments where you look at glucose uptake. And again, that's just measuring kind of their activity, getting a feel for their physiology. So this table is just trying to give you some ideas of different things you can do with those samples. And there's definite advantages and some disadvantages depending on which technique you pick. So I guess one of the big take-home pictures for this is, whenever you design an experiment in microbial ecology, you have a lot to think about in the sense of course, remembering the difference in size between you and a microbe. is a huge thing to think about for how are you going to sample. And then what technique should you use? You really do need to be aware of what the pros and cons are and what that technique really does allow you to answer in terms of, you know, some techniques can give you some great data, but they might not really get at what you're most interested in. So you have to be very thoughtful up front when you're designing these experiments so that you're actually going to get data to answer questions that you really want answered. So again, just emphasizing the viable plate count technique. It is publishable. I like doing it. I do it almost all the time when I'm running experiments. That's part of the process. But we know it underestimates. But we all know that. So as long as we're all aware of that, it's fine. And so obviously it's hugely selective because you're the one picking the media recipe to make those agar plates. So depending on what recipe you use, you're definitely going to skew who shows up on those plates. And of course, only living microbes are going to grow. Obviously, the ones that you plate out that are dead, they're just going to stay dead and you'll never know that they were there. There's another technique I haven't showed you in the lab, but it's called most probable number. I'll show you a picture of that in a minute. And it's really straightforward. You don't get to see any of the microbes though, so I don't do this one that much. But it's a quicker way. The most probable number is a quicker way to get at population levels because you don't have to wait for the colonies to grow up. So I guess in that respect, the MPN method is quicker and a little bit quicker. You have to let them grow up in a broth, but I'll show you in a second what I mean. So, you know, these viable count processes onto agar plates, that's a spread plate technique that I've taught you. So spread plate is what we use for the viable count. Pour plate is another thing, but really I don't see that being used all that much anymore. I think it's gone by the wayside for the most part because there's better techniques to do. Basically the pour plate, instead of spreading your serially diluted sample onto the agar, you mix it into the agar and then you pour that into a plate. So then the microbes are in the agar and then they're also on the agar. To me, it doesn't really make sense to do it that way because there's other ways. It's not as accurate when you do counts. It's kind of something that you'll see in the older literature, but I would never really recommend it. The spread plate is much better. So serial dilution, the most probable number is a serial dilution idea. So that's what they're showing here. You have your sample that you add, so say it's a soil sample that you popped into some broth and you're growing it up, and then what you do is add in 1 mil into the first tube, and it's usually 9 mils into all these tubes. You put 1 mil of your sample, and then you serially dilute like what we've done in lab, and then you incubate these tubes. And what you're looking for is where in this dilution scheme do I see no growth? And that's what you want. You want to see, okay, here at 10 to the minus 6, I see no growth. The rule of thumb here that microbiologists have agreed to is that the last tube showing growth, so it's the 10 to the minus 5, they're saying that one must have developed from 10 or fewer cells, because when you try to take a sample from that and go into the next tube, you're getting nothing. And so the thought is, okay, so then the previous tube must have had less than 10 or fewer cells to end up with the next tenfold dilution showing nothing. So in this case, the most probable number would be whatever, it'd be 10 to the 5th, essentially, of organisms showing up. So it's a way to estimate your population, but you don't really have to do a viable count. You can just work with the tubes. And so it's a little bit quicker. I don't really use it that much because I like to see who's showing up. I want to see the colonies. So I usually do a spread plate which you've already done. This is a spread plate idea. You've seen this already. The pour plate is being shown on the bottom, where you mix your sample into the agar and then pour it in and incubate it, and then you get some bacteria in the agar and some on the agar. And I guess when they designed this way back when, they thought, well, if microbes are in the agar, that means they can handle lower oxygen, because if you grow in an agar, you're not getting much access to oxygen. So it gives you some information about their oxygen requirements, but there's way better ways to get at that. And so I find that the pour plate pretty much, it's outdated, and there's way better ways to do that. So, so I would not recommend it. Spread plate definitely is good. Pour plate, no, not so much. But you do see it in some of the literature and not some of the older literature. So I just wanted to make you aware of it. One other technique, colony hybridization. So in the microbial world, what you would do, so colony, they're saying you need to isolate colonies on a plate. And then hybridization means somehow you are bonding something to it, you're hybridizing. So basically what this is you do typically a serial dilution, spread plate technique, incubate the plates, get your colonies to show up. Then what you do is you take this special membrane and you very carefully, it's cut to the size of the Petri plate, so you lay it on top of all your colonies on that plate, and you essentially create an imprint, or you transfer now all the microbes onto that membrane. So it's just a way to get a faithful representation of that plate, all the colonies on that plate, you transfer to a membrane, and then you start working with the membrane. So now you have this membrane that has all those colonies on it, and it's a perfect match to your agar plate colonies. And then you use that membrane and you run your experiment, your hybridization assay, with a DNA probe. So if I want to ask, okay, I have all these colonies on this plate, I want to know which of these are antibiotic resistant microbes. So then you could take a DNA probe that has the DNA sequence that codes for antibiotic resistance of whatever one you're interested in, and you run the experiment on the membrane. So it's being shown here. So here's your agar plate with your colonies. You press the membrane down to create a perfect copy of it. And then you work with this. You bathe it in a solution that helps gently release the DNA, but not obliterate everything. And then you do some more steps to kind of stabilize the DNA in place, pre-hybridize, and then you add your DNA stretch that has a probe on it. The old way to do it was through autoradiography, but they can do fluorescent techniques now. And then what happens is when you process the membrane after you've added your probe, this is autoradiography, you can see all the colonies that turned dark are the ones that took up the probe. So there was a complementary copy of your probe to the DNA of that microbe, and so they popped, and you can see that all of these microbes, maybe, they don't say what they were actually looking for here, other than that it was Burkholderia, but maybe it was antibiotic resistance. Maybe all these microbes had the gene for antibiotic resistance that your probe was able to complementarily bind to. I can't say that word. So again, you can go back to your plate and say, okay, it looks like, these 3 top colonies do have the antibiotic resistance. Well, you still have a copy of the plate with the actual cells on there. So then you go back to your plate and pick them off, and you can start working with them if you want to. And so this is a pretty simple, tried and true method, pretty elegant in its simplicity, really. And it allows you to maintain live colonies still as backups. So you can go back to them later and start working with them when you find out which of those colonies have your gene of interest. So pretty straightforward, pretty slick. Okay, I'm going to update the slides. There are a couple of slides that have this reading I did not really assign to you, but I just took very select sections of it to just get across the point of how they're using some of these methods that we've been talking about. And this is in an ag field where they were looking at the input of different fertilizers, and biochar is a really interesting amendment that you could put into soil systems that really stabilize. It allows for microbes to have a house, essentially. Think of it like a home for microbes. It's a physical substrate you put in, and the microbes love to attach to it, but it really doesn't break down very quickly. So literally it can stay in the soil for thousands of years. And so it's been used in South America for thousands and thousands of years. They've known about this. We're starting to use it more in ag fields now. This is just the abstract. The main point I want to get across is that they were using phospholipid fatty acid techniques. So here, this was published this year. I just wanted to show you how they might use some of these techniques we've been learning about. And they were looking at using the phospholipid fatty acid to look at gram-negative bacteria, gram-positive bacteria, mycorrhizal fungi. And so they were able to, let me show you a couple of graphs here. So this is total phospholipid fatty acids, and they were just asking the question, when we do different amendments into the soil, what happens to the microbe community? And so you can see the bars aren't all the same across the treatment. So NF is no fertilizer, nothing was added into the ag field. Biochar at different amounts, and then organic nitrogen at different amounts. And you can see that the population levels of the microbes, by tracking the phospholipid fatty acid amount, you can see went up in the organic nitrogen and not so great in some of these others. All right, so then they were able to tease it apart because the phospholipid fatty acid is a way you can actually tell the difference between gram-positive, gram-negative fungi, eukaryotes they even looked at. So again, by using this technique, they are able to generate some really good data on, okay, let's look at the entire microbial community, and then let's tease it out by the different groups of microbes and see what's happening when you have these different amendments put in, different treatments put into the soil. So I don't want you to get lost in like knowing the details of this study. It's more of what technique did they use and how powerful it can be. You know, a good example of what kind of data you can get from this and the level of detail. Again, just looking again at all the different treatments, breaking out all the different types of microbes. And then going back, it's an ag field, so they were looking at pepper plants and looking at pepper yield. So again, showing that the idea of, it was a 2-year study, so the dark bars are the second year, and you can see that the input of biochar or the organic nitrogen really did allow for some nice high populations, which is what they hypothesize. If we have high populations of diverse microbes, that's probably a pretty healthy agricultural system. So that's one example of how we can use some of these methods to answer some questions. This is the other one I assigned, but really don't get lost into the methods. This one, though, is they were using DNA sequencing essentially, and they were looking at 16S ribosomal RNA sequencing, which is a hugely popular technique. So again, I just wanted to use this as a way, it's a 2020 publication, use it as a way to show you how we're using some of these methods in the real world arena of research, okay? And then this one I just mentioned the other day. This is just if you're curious. The big picture idea, though, I think the title kind of says it all, rare rather than abundant microbial communities. So I do want you to keep this in mind. You don't have to read this paper, but I just took little snippets from it in this slide and the next slide, just to get you thinking about the idea. This is kind of a new idea that we can now test because we have the tools to do it. Whereas before we always thought about it, but we couldn't answer it. So we're finally able to get at this idea, how important are the lower populations that we see in communities? And in this particular case, they said they think they're really important and could be keystone species. Okay, so again, there's just a couple slides on that. And really the title kind of says it all and the overall statement here. Okay, so these are some of the methods I wanted to expose you to with what's going on in microbial ecology. So now that's pretty much all I'm going to say about it right now. And then we're going to shift into the next topic tomorrow when we all zoom in together, okay?
Just curious, what is it going to be today? So,
What was the question?
like the next topic.
Oh, it's biogeochemistry. So biogeochemistry, I'm going to have to shorten that quite a bit because we're behind schedule. So that one's going to get shortened because we have to have enough time to get through all the different microbe organism interactions, which is a lot. So biogeochemistry is the next one, which kind of your Winogradsky column is getting at that idea.